So welcome back to molecular biology. This is session 4. So the learning objectives for this next session are to describe the different types of genome, transcriptome and protein analysis available for molecular biology analysis. The second objective is to describe the molecular theory behind each of the methods of genome, transcriptome and protein analysis. And the third objective is to analyse genomic, transcriptomic and proteomic data. So this is the introduction. So genomics, proteomics and metabolomics generally is known as omics technologies. So genomics is the description of genetic information, whilst proteomics is the description of proteins. Metabolomics is the kind of qualitative and quantitative analysis of all low molecular weight metabolites of a cell or organism and their dynamics in the kind of biological systems. Now, these technologies will kind of allow the analysis and characterization of biological systems, and thereby kind of opening up new perspectives for ethnobotanic and medicinal plant research purposes. So what is bioinformatics? Well, bioinformaticians are concerned with deriving biological understanding from a large amount of data with kind of specialized skills and tools. Now, bioinformatics is actually really kind of a broad discipline. It kind of encompasses many aspects of the kind of omics technology. But it also kind of includes things like structure informatics, comparative genomics, machine learning, and image processing. The focus primarily is on the kind of sequencing data, and there are kind of broad applications across biology, looking at things like variant detection and genotyping and gene expression, as well as protein DNA interactions such as ChIP-seq. So for a biologist, bioinformatics means kind of different things to different people. So for a biologist using computers or the other way around. And bioinformatics is kind of basically the combination of biology and computers. For a bioinformatician, they have a different kind of belief or view of the world. And it's the use of kind of a computational tools to kind of expand and organize and analyze scientific data. So it kind of means different things to people. In fact, now the cost and availability of the kind of next generation sequencing is kind of coming down. In the past, when we used to kind of sequence DNA, you would often produce a picture like this. Essentially, this is a film from a sequencing reaction. And we would sit and kind of work out each of the kind of the bases in our sequence. Now we've got sequences such as the MISEQ. There's also different types of sequences which are kind of benchtop. And now actually we have miniaturized sequencing. This one's known as the Minion, which is kind of a portable real-time device which can be used for DNA and RNA sequencing. In fact, now we can actually, with this tiny little device, we can generate more than 30 gigabytes of DNA sequence. And in fact, the current record is now about 43 gigabytes. So there's kind of like a number of advantages of sequencing. So you've got construction of a sequencing library and also clonal amplification to generate sequencing features. So there are no in vivo cloning, so transformation and colony picking. So it kind of reduces the time that we spend in the lab checking our kind of colonies. And we can use sequencing now to kind of get that information quite quickly. There is array-based sequencing, and there's actually a high degree of parallelism that than the normal kind of capillary-based sequencing, which traditionally the Sanger sequencing would use the capillary method. Now we can use something called next-generation sequencing. So genomics was kind of originally meant as the analysis of the whole genome, and now it's kind of commonly refers to a kind of large-scale and high-throughput molecular analysis of multiple genes and gene products or their regions of genes. Whilst the transcriptomics is often kind of included in the term of genomics and depicts the expression level of genes, and this is for both the techniques that are kind of DNA sequencing or DNA fingerprinting and DNA barcoding. So genomics is looking at obviously the DNA that's kind of we find in the nucleus, so the synthesis of this DNA. Whereas the transcriptomics is looking at the messenger RNA, so it's the expression, so it's the transcription of that DNA into RNA, and we're looking at the kind of the quantity of those RNAs, which kind of gives us a kind of a feel of the expression of genes. To understand better about whether or not those genes are expressed, because some genes are only expressed at the messenger RNA level, and these may act as something called long coding RNAs, or they may be acting as microRNAs. So to really get a feel of whether or not the gene is actually being really expressed as a protein, we need to do proteomics. And proteomics is kind of looking at the proteins that are produced by translation by the ribosomes. So the genome is kind of really made up of this entire complement of genetic material of the organism, a virus or even an organelle or a haploid set of chromosomes in the eukaryotic organism. The whole genome is essentially the whole complete genome set of the organism, and whole genome sequencing is essentially this is the kind of laboratory process where complete DNA sequence of the organism's genome at a single time. The term sequence analysis means that it's kind of subjecting the DNA or the peptide sequence to sequence alignment, and sequence databases and repeated sequence searches or other bioinformatic methods using a computer. The sequence analysis in bioinformatics is an automated computer-based examination of characteristic fragments, for example, of a DNA strand. And so the whole genome analysis equals the whole genome sequencing, as well as a combination of bioinformatics. So genomic analysis is kind of the identification of the measurement or the comparison of genomic features such as the DNA sequence, the structural variation, also the gene expression or regulatory and functional element annotation kind of at the genomic scale. Now, there are a number of methods for genomic analysis, because these are typically require high throughput screening or microarray hybridization and bioinformatics, which we'll kind of discuss later. This is really kind of a snapshot of the kind of sequencing history. So Alan Maxson and Walter Gilbert developed the chemical method that we know as DNA sequencing back in 1976. Between 1976 and 1977. Since this method was technically complex and the use of extensive hazardous chemicals, it kind of fallen out of favour. So back then they would use radioactivity to label the nucleotides. And Sanger-Coulson actually then developed the chain termination method in 1977. And this only was to be used for fairly short strands of DNA, anything between 100 and 1,000 base pairs. And so longer sequences had to be subdivided into small fragments. So bacteriophage FX174 was actually the first genome to be sequenced back in 1977. It has 11 genes and with a size of about 5,368 base pairs. Now, this is a viral genome which is kind of smaller than T phages and are kind of polyhedral. and this was actually done by Norman and Baker by staining method with the use of the Sanger method of shotgun sequencing. Haemophilus influenza was actually the first bacterial genome to be sequenced back in 1995 by Craig Venter. This is a gram-negative bacteria with 1.8 billion base pairs. And the first nearly complete human genomes sequenced were from Craig Venter, James Watson, Yoruba, and Sol-Jin Kim. So why is it important to sequence the whole genome? So we get information about the coding and non-coding part of an organism, but we find out about important pathways in microbes. It's also useful for evolutionary study and also if we want to compare species. It's for more kind of effective personalized medicine, so we can understand why drug works in person X but not in person Y, and so we can design our treatments to treat the person. The identification of important secondary metabolite pathways, so for example in plants, so for the production of potentially drug compounds or drug-like compounds. For disease susceptibility prediction, so that's kind of based on gene sequence variation. So we can make a comparison of kind of healthy individuals and people with, for example, lung cancer. So for the stages of genome analysis, so first we kind of start off with is the genome sequence assembly. What we need to do then is to identify kind of repetitive sequences, which kind of helps us to kind of mask out those areas. Using the repetitive sequences, then we can this can provide us with a gene prediction, and so certain genes will have certain repetitive sequences, and so we can have kind of a model for each of the genomes. The genome annotation is the process of attaching biological information to the sequence. The metabolic pathways and regulation, this is to kind of find missing genes. We can also do protein 2D electrophoresis, and so with this we can actually detect for the translational products, and we can detect for differences between samples. We can also do functional genomics, gene location, gene map, self-comparison of the proteome, comparative genomics, as well as we can identify clusters of functionally related genes. Also, we can do evolutionary modelling to analyse chromosomal arrangement, duplications, and predictions that can be made. For gene prediction and recognition, it is easy for prokaryotes because there's just one cell, and so potentially just one protein. Whilst for eukaryotes, they are kind of multi-cell, and it's much more difficult because there's one gene but many proteins. And so it's very difficult for humans because we have exons, short exons, which kind of separate by then kind of non-coding long introns. And so gene recognition is by kind of sequence alignment. So if we look back in the past, so since like the Human Genome Project, which started in about 1990, it took around about 13 years to complete, and arguably it was this kind of sequencing of express sequence tags known as ESTs. Now it cost around about 2.7 billion. In 2015, Illumina actually announced a landmark of $1,000 to sequence a human genome, and so the cost of sequencing has come down immensely. If we take a look at the human genome data, for example, there's 3.1 times 10 to the 9 base pairs in the human genome. Now, the difficulties are that small genes are quite hard to identify, and some genes are actually rarely expressed, and so they do not have actually normal codon usage patterns, and so are actually difficult to detect. If we take a look at the human genome data, so this kind of gives you an idea of how the cost of sequencing has come down. So between the periods of 1990 to 2003, the turnaround time was about 5 years, and the cost per genome was about 3 billion. Between 2003 and 2009, it took around about 6 months to generate data for a genome, and this was a cost of about 300,000. Between 2000 and 2014, in less than 1 month, we could turn the sequencing data around, and for exons, it was cost of 3,800, and for whole genome sequencing, 20,000. From 2015, it now only takes 15 minutes, and the cost of the genome is about $1,000. So it's not just about, the genomics is not just about genomes. We can also do things like single-cell RNA sequencing. And single-cell RNA sequencing essentially examines the sequence information from individual cells, which is kind of optimized with the next generation sequencing technologies, and then provides us with kind of a higher resolution of cellular differences and a better understanding of the function of an individual cell in the context of its microenvironment. Now, we can have a look at this website from EBI, and they actually update this kind of periodically. Example, we can have take a look at the Atlas of Human Blood Dendritic Cells and Monocytes, where they've actually sequenced individual cells. So, data availability. So, there are a number of kind of websites that we can take a look at. For example, the 1000 genomes, and we'll have a look at this in the session. Also, the ENCODE RNA dashboard. This is also a really good resource. And also, this is from Patrick, looking at antibiotic resistance data. Now, all this data is kind of freely available to the public. Moving on to transcriptome. So the transcriptome is a complete set of transcripts in a cell and their kind of quantity, and it's for a specific developmental stage or physiological condition. So by understanding the transcriptome, it's essential then for interpreting the functional elements of a genome and revealing the molecular constituents of cells and tissues, and also for the understanding and development of disease. So transcriptomics is essentially the study of RNA in any of its forms. So the transcriptome is kind of a set of all the RNA molecules. This includes things like messenger RNA, microRNA, ribosomal RNA, tRNA, and other non-coding RNAs that are produced in one or a population of cells. So transcriptomics can be applied to kind of a total set of transcripts in a given organism or to a specific subset of transcripts present in a particular cell type. So unlike the genome, which is kind of roughly fixed for a given cell line, excluding mutations, the transcriptome can vary with external environmental conditions. Since it includes all of the messenger RNA transcripts, so this is the RNA which is kind of is going to be translated in the cell, the transcriptome kind of reflects the genes that are being actively expressed at any given time. And so the exception of messenger RNA degradation phenomenon, such as kind of transcriptional attenuation. So the study of transcriptomics is also kind of referred to expression profiling. And this looks at the expression level of messenger RNAs in a given cell population. And it's often used in high throughput technologies based on DNA microarray technology. So the aim of the transcriptomics is one, to catalogue all the species of transcripts, including messenger RNA, non-coding RNAs, and small RNAs like microRNAs. Also, we can determine the transcriptional structure of genes in the terms of their start sites, the 5', the 3' ends, as well as the splicing, alternative splicing patterns, and other post-transcriptional modifications. We can also quantify the changing expression levels of each transcript using the development and under different conditions. So the technologies that are around that we can use to understand things like these are kind of related to obviously to the genomics. We have kind of hybridization based approaches. So, for example, we can use fluorescently labeled cDNA with custom-made microarrays. We also have commercial high-density oligomer microarrays. We also have sequence-based approaches, so things like Sanger sequencing of cDNA or EST libraries. We have something called SAGE, which is serial analysis of gene expression, CAGE, which is a cap analysis of gene expression, and MPSS, which is massively kind of parallel signature sequencing, all different types of technologies. So the microarray method, that's used to check for gene expression. And microarrays are essentially DNA chips which kind of use thin glass microscopic slide which has a kind of maybe a silicon chip or a nylon membrane. What happens is we have like reference genes which are kind of immobilized or spotted and then synthesized in situ on this kind of small space on the glass slide. And it works on the principle of hybridization, so the attachment of the messenger RNA, which is then converted to cDNA in the concerted cell with an immobilized cDNA and oligonucleotide sequence, which is present on the array slide. So the advantage over SAGE is that there's a rapid evaluation of the comparison of the transcriptomes that can be achieved by running them simultaneously on identical arrays and checking hybridization patterns of the two. We can then refine the results, which then can be achieved by using messenger RNA that is bound to a ribosome in the cell. Now, the ribosome-bound messenger RNA can then give part of the transcriptome that is kind of actively being used in the protein synthesis. If we can compare microarray and DNA chip, the microarray uses PCR products or cDNA of genes of interest, and these are spotted onto the surface of the glass slide or nylon membrane, whilst the DNA chip uses oligonucleotides which match the positions within the gene of interest, and the mixture of oligonucleotides are then synthesized in situ on the surface of the glass slide or the silicon membrane. So by studying the transcriptome, this can lead then to various applications. So we can look at transcriptomes of stem cell, cancer cells that can be studied by researchers to understand the cellular differentiation or carcinogenesis. We can also look at the transcriptomes of human oocytes or embryos that can be studied to understand the molecular mechanisms and signaling pathways in embryonic development. And this also can be used in biomarker discovery. RNA-seq. So these are essentially transcriptomics, which kind of replaces the hybridization of a nucleotide probe with a sequencing individual cDNA, which is then produced from an RNA, target RNA. So the emerging methods for these fully quantitative transcriptomic analysis have the potential to overcome the limitations of the microarray technology, and there are ongoing discussions about whether sequencing approaches may replace microarrays in the middle or even the short term. So the microRNA profiling involves a number of steps. This is an example of a microRNA profiling platform available from HTG Diagnostics. So what we do is we start off with the sample preparation. So normally we have to use a small sample input. And the sample can be anything from tissue to plasma serum, as well as like cells or purified RNA. And there is lysis chemistry and no nucleic acid extraction. Now, this step takes around about 4 hours with kind of 30 minutes hands-on time. Afterwards, there is a kind of library preparation, which involves first the target protection. So it's kind of a fully automated system, which is kind of related to the HTG diagnostic platform, where they use nuclease protection assay. Afterwards, they do like a library amplification. So here they add both barcodes to which are then kind of tagged, and then they add the adapters, and then they're amplified. The next stage is the library quantification and normalization, which involves the library cleanup to remove any excess primers, and then the library quantification. This then is normalized and then is fed into the next generation sequencing, and then afterwards it's the data analysis. Have a look at the YouTube video on the slide. So for next generation sequencing, it's kind of recently is a kind of a number of like faster and cheaper sequencing methods have been developed. And in fact, in October 2006, there was the X Prize Foundation, which was working in collaboration with the Craig Venter Science Foundation, which established the Archon X Prize for Genomics, which was intended to award $10 million to the first team that could build a device and use it to sequence 100 human genomes within 10 days or less, with accuracy of no more than one error in every 1 million bases sequenced, with sequencer accurately covering at least 98% of the genome and at a recurring cost of no more than $1,000 per genome. It had an error rate of 1 in 1 million bases, out of a total of approximately 6 billion bases in the human diploid genome, which would mean about 6,000 errors per genome. In clinical use, such a predictive medicine currently have 1,400 clinical single sequencing tests are over in August 2013, which have been cancelled. So currently there is a developing method that will kind of sequence the entire human genome for $1,000, and this kind of allows personal genomics. One of the most widely used methods involve pyrosequencing. So this is kind of biochemical reactions invented by Narin and Renaji in 1996, which is kind of massively, it's kind of parallel microfluidics technology. So microfluidics is kind of a up and running application, and it was invented the 454 life science company, and they combined the technology, which is called now 454 sequencing. So now we will look at the next generation sequencing. So it's an array-based sequencing. The sequence, you can sequence full genome of an organism in a few days at a very low cost. As well as we can produce high throughput data in the form of short reads. Now there are different systems available on the market. We've got the Illumina, the ABI Solid, the Roche 454, FLX, and the Ion Tolerant. So the capacity of next generation sequencing is greater than 1 gigabase per run, and there is a drastic decrease in costs per genome. Sequencing of the whole bacterial genomes in a kind of single run. You get sequencing of genomes of individuals. We can get information about metagenomics. So this is sequencing of DNA extracted from the environmental samples. And so we're looking at rare variants in a single amplified region, in tumours or in viral infections. And transcriptome sequencing is kind of total cellular messenger RNA, which is kind of converted to cDNA. So this is actually a paper, quite an interesting paper, looking at metagenomics, and here they're kind of looking at the recovery of nearly 8,000 metagenome assembled genomes, which kind of substantially expands the tree of life. So have a look at this paper. So where can we use genomic or next generation sequencing? So there are a number of applications. So it can be used in oncology. So we can determine the preferred therapeutic agent for each tumour. We can ascertain which patients are most likely to benefit from a given therapy. For molecular pathology, there are disease-specific tests. So the cost of a single test anything between $100 to $5,000. We can have individual test validation and performance, as well as for medicine. Human genomic sequence in the public databases allows rapid identification of disease genes by positional cloning, and also inherited diseases can be identified. Next, we're going to move on to proteomics. So proteomics essentially is kind of analyzing the proteins, okay, identifying the proteins, okay. So these can be proteins or peptides, okay. So we're going to kind of look at how proteins are kind of analyzed. So mass spectrometry is kind of the analytical instrument that we use in proteomics, and it's used for all types of chemical analysis. But we can also measure the elemental composition of matter. We can analyze structures of organic and inorganic biological molecules. We can find isotopic ratios of atoms in the sample, and it's also for the detection of poisons in a sample. So there are various types of spectrophotometers that are available, which allows us to separate the ions according to their mass to charge ratio, or simply on the basis of mass analyzers. So they are, for example, 1, magnetic deflection mass spectrometer, 2, electrostatic mass spectrometer, 3, time of flight mass spectrometer, and 4, quadrupole mass spectrometer. So we've got an example of the magnetic deflection mass spectrometer. So we start off with the source. So these are this is like the ion beams. And for most mass spectrometers, you always have a magnet which helps to deflect the signal. Okay, so here you would inject the source, which then would be heated up in a chamber and then ionized, and then the molecules then would move through this ion beam, which then would be deflected in the magnet. The ones which are less deflected are the kind of heavier atoms, and the ones which are lighter can be kind of separated into different ion detectors. So in the mass spectrometer, the sample is analysed. It's kind of first bombarded, so with the electron beam to produce ionic fragments of the original molecule. Now, these ions are then accelerated in a high vacuum into the magnetic field, which then deflects them into a circular path. The deflection for the light ions is greater than that for the heavy ions, and the ion stream then separates it into beams of different molecular weight. And a suitably placed slit allows the beam of a selected molecular weight to kind of pass through to a collection electrode or detector. We also have it's called the electrostatic mass spectrometer. It's quite similar to the one that we looked at before. Here, again, the sample is injected, it's ionized in like a vacuum, and then it kind of passes through this electromagnet, and you get like deflection, and then it moves, and then these then move then towards the detection. So the electrostatic sector kind of analyzer consists of kind of two curved plates of equal and opposite potential, and as the ion travels through the electric field, it's then deflected, and the force on the ion due to the electric field is then equal to the centripetal force on the ion. Here the ions of the same kinetic energy are focused, and then the ions of different kinetic energies are then dispersed. And the electrostatic and magnetic sector analyzers that when employed individually are then single focusing instruments. And when both techniques are used together, it's then called a double focusing instrument, because this instrument has both the energies and the angular dispersions then are focused. The time of flight. So time of flight mass spectrometry is kind of a method for measuring the distance between a sensor and an object. So it's based on the time difference between the emission of the signal and its return to the sensor. So after kind of being reflected. So here we have our sample, which is ionized. We have the laser, and then here are is the slit where you've got the electric field generator. So as the ions kind of enter through, the smaller ones are going to move quickly. So essentially, this is an example of a time of flight mass spec. This tube here is actually where the ions kind of move up, and the small ions will move quicker, and the larger ones will move slower, and then it kind of moves to this detector plate. So the TOF mass spec, the ions are different mass charge ratio, are kind of separated by the difference in time they take to travel over an identical path from the ion source to the collector at the detector. Here the ions are then generated by bombardment of the sample with a brief pulse of electrons and the secondary ions or laser generated photons. The ions are accelerated by the electric field and they kind of enter this field-free drift tube. And the ions then enter the tube with the same kinetic energy and ion velocity, which will vary inversely according to mass. So the lighter particles will arrive at the detector before the heavy particles, and the ions are then separated in the drift tube according to their velocities. So the advantages of TOF is that it's a kind of high-speed operation, so you can get information very quickly. It has the ability to record the entire mass spectrum at one time. You get accuracy only, which depends on the electronic circuits. The disadvantages of TOF are that there is kind of poor resolution due to the display on the oscilloscope screen. For the applications of TOF's mass spec, there are kind of qualitative analysis and also quantitative analysis. So for qualitative analysis, we can look at the molecular weight determination and also the structure determination of a substance. For quantitative analysis, we can use this in kind of biotechnology, for the analysis of proteins and peptides, as well as for the analysis in pharmaceuticals and drug discovery. Finally, now the quadrupole. So the quadrupole essentially is these kind of four parallel cylindrical rods, and these kind of act as electrodes, and this is kind of an example of what a quadrupole looks like. So as I said, there are four parallel cylindrical rods which serve these electrodes, and the opposite rods are then connected electrically. So you have one pair which is attached to the positive side of the variable DC source, and another pair that are attached to the negative side of the DC source. You have a variable radio frequency of AC potential of about 180 degrees out of the phase, which is applied to each pair of the rods. And this kind of results in an AC potential superimposed on the DC potential, and this creates an electric field. The ions are then accelerated through the electric field between the rods. The AC and the DC rod voltages are increased simultaneously with ratio being constant. All the ions without specific mass to electron strike rods then become neutral. Only ions having limited range of mass to electron reach the detector. So now we're going to focus on how to analyse genomic data. So the picture you can see on the screen is essentially a chromatogram of sequencing data. And each of these peaks represents a different nucleotide. So for example, the black peak is G, the blue peak is C, and the green is A, and T is the red peak. So what we can do is we can actually work out the sequence data. Okay, so you can have a go at trying to do that. So if you had a go at trying to work out the sequence data, what you would find is that this peak here is a G, and then you've got a C, 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 and then a T, a G. And so you can read off the chromatogram. This is particularly useful if, for example, you get a sequence where the software is unable to kind of decide what peaks are present. So you may have some little bit of noise, or the sample is a little bit dirty. And so you can actually have a look then at the chromatogram and determine what the sequence reads. So we could take our sequencing data like we saw before, where we had to go kind of reading off the chart. Now you can actually see on this sequence data, in this chromatogram, is that the beginning of the sequence is fairly noisy. So you can see like there are peaks where it's very difficult to kind of analyse the data. So in fact, it's probably best to start the sequence from about 30 base pairs. So we can take our sequencing data and put it together And this picture actually shows you where the sequencing is not aligned. So you can see that there are the grey boxes where it's been aligned with each other, and in the white boxes there is less homology. If we take the same sequence and then align it, then we start to see some differences here. So now we can see that there isn't those gaps, and we can see that there are kind of areas where there is kind of like high homology, and there's kind of like a variance of homology. So I'm going to share you some kind of websites which are particularly useful, and there's a couple from Broad Institutes. So we've got the Broad Institute is like data sciences, and also this website here is looking at cancer cell lines. This website is quite interesting because it measures the messenger RNA expression in like thousands of cancer cell lines, which we can then download the data from. There are other websites which are available known as the Protein Atlas, which gives us an idea of the expression profile of proteins in the body in different cell types. And also we got the Human Cell Atlas, which is kind of a recent thing, which is kind of still being developed. So on the next few slides, there are kind of further details about the technologies which have been kind of discussed in this session.